harder and harder as you move on with your classes. Okay, let's get going so I could finish on time. Today's session is about protein synthesis. And we'll try and basically describe to you, as we did before, just as a reminder, the genetic code. And we'll talk about some DNA mutations and how one nucleotide, could mess up the whole protein, could cause a syndrome, and would cause a lot of damage. So we'll give you some examples. And I'll tell you what kind of questions to expect in the TBL, in the exams, and so on as well. And I do require you to basically understand what are the components that are needed for translation, and the steps of translation, and also we'll talk about co- and post-translational modifications that do occur, and we'll describe some conditions also associated with impairment of post-translational modifications, especially SCARV, and that's what we'll discuss as well. Okay, as we discussed before, information flow basically goes from a gene to an RNA, mRNA. If you notice here, we have DNA template strand. You have to know the name. This is the template. So the mRNA is complementary to the template strand. Before I forget, I did pass Lippincott's Q and Answer book to your students who were supposed to post it in your groups. And I do encourage students actually to look into these questions because they can get you familiar with the kind of questions you will be expecting in the final. And these are wonderful. And the best thing about this book is that it classifies the questions according to the subject. So when you're talking about RNA, you have RNA questions. When you're talking about protein, protein questions. If you look into other exam review questions, you'll see it all over the place. And I did suggest to some of you to form a group where they can take some of these USMLE exam review questions and classify the questions accordingly. So that when you're studying protein, you have questions about the protein. When you're studying, I understand this is going to take some time, but it will help you in terms of getting more focus rather than general questions that you'll find in these exam review question books. Okay? The reason I'm saying this because it's not going to be simple, straightforward questions in your exam. I'm telling you right now. I'm not going to come and say, well, what's the strand for this or that? It has to be tricky in terms of shows that you understand the kind of questions and what to expect. It all has to be critical thinking. It's not going to be recall. We will give 20% of the questions will be recall questions. 80% will be critical thinking, as simple as this. And we are discouraged by the higher admin. No recall questions because we have to classify each question and they count for us what's the percentage of recall questions. And that's how they evaluate and validate each exam. Okay, let's get back and start moving. So we have the mRNA. If you notice at the mRNA, we have what is known as triplet code. And we can see that 3 nucleotides. This is high school information that you already know, triplet code. And then you can see that each 3 nucleotides code for 1 amino acid, and that's when we have translation into a full protein. Now everyone is going to ask me, are we going to remember this table? The answer is no. I do want you, the back there, I can see that you're enjoying your time there and chatting and chit-chat. So I do need you to know the stop codon. And it's easy to remember. You have UAG, then flip it, UGA, and then UAA. These are stop codon and the start codon, AUG. These are the ones that I want you to remember. So AUG codes for a methionine, and that's your start codon, usually. In eukaryotes, yes, it's the start codon. In prokaryotes, we may have some other start codons. Okay? If you notice here, we have these stop codons, or what they're known as nonsense. So always remember, stop codon, nonsense. Then we have two amino acids, methionine and tryptophan, that have only one coding triplet. These are specific. Each codon codes for one amino acid. That's why we refer to them as unambiguous. Universal, all strains. You go to cats, you go to yeast, you go to bacteria, it's the same triplets, coding triplets. That's why we call them universal. They are degenerate, meaning they're redundant, meaning that you can see, for instance, for proline, 4 triplets that code for proline. That's why we call them degenerate or redundant. There are around 64 different triplets. So what happens when one nucleotide, just one, changes? You could end up with what is known as termination codon being established as nonsense. So stop codon, meaning termination. That's why we call it termination or nonsense mutation. Meaning that if you have a long protein, if there's a mutation where you end up with a stop codon, that protein could be non-functional. But we already took an example, the B100, B48, I'm sorry, and we showed you that how the termination codon could code for a protein that is excreted by the intestine versus the one by the liver. As simple as this. But at the same time, we could end up with nonsense mutation stopped. So you have basically partial protein, so it could be non-functional, and it is destined for destruction. As simple as this. We have another mutation. These are common questions, just to let you know. We ask what kind of mutation you're seeing there if we switch this to this. So at least understand the nonsense mutation. We have the missense mutation where you end up with a different amino acid. So this is for proline, this is for serine. Just by mutating what? The first nucleotide from U to C. So we end up with different amino acid being incorporated. And we'll take some examples of these mutations. Finally, we have what is known as silent mutation, meaning that if there's a mutation, nothing happens, the same amino acid. This is why we have polymorphism. Your DNA is different. Why? Because we have silent mutations. How often do we get silent mutations? You're sitting right now, you have silent mutations happening in your bodies right now, in your cells. Why? Because cells multiply, proliferate, and they're not basically 100% error-proof. So what happens? You end up with mutations. This is why we have polymorphism. Every person has his own DNA. Because during replication of the cells, you get basically some errors, some mutations right there. Lucky for us, 99% of the DNA is nothing other than junk. So the mutation chance happening basically in these, in the 99% is higher. That's why we don't have that much effect on our proteins, as simple as this. So we have silent mutations, we have basically nonsense and missense mutations. We have another term that you should be aware of. It's called frame shift mutations. Why do we teach you frame shift mutation? Because you're going to see it in textbook. You're going to see it in the questions. There is a frame shift mutation, meaning either addition or deletion of 1 or 2 nucleotides. If it's 3 nucleotides, that amino acid being coded for by that triplet is basically going to disappear. Or if there's insertion. But if there's one or two, what happens? We have completely different protein. Could be junk. Even though it's synthesized, it's dead protein. It's useless. So we have example, we have serine, proline, methionine, alanine. So if you add one nucleotide, you're shifting the reading because we're reading every 3 nucleotides, triplet codon. So what happens? So you end up, if you insert one, for example, this is just an example. Serine, serine, tyrosine, glycine. Completely different protein, could be junk. And it's not going to be functional. Is it the same amino acid? If it's the same amino acid, you're okay. It's, yeah. The problem, if it starts coding for another amino acid, we're going to take an example and we'll discuss what happens. Just one mutation changes the amino acid, so we have sickle cell anemia, simple as this. I'll give you some examples. So just to show you what we mean by frame shift, we just inserted one nucleotide and all of a sudden we have completely different amino acid. That amino acid, this protein is basically useless and will be destined for destruction. Yes? Can't hear you, can you? So if you look into the other example, if there's a deletion of one nucleotide, just one, again, we have serine, leucine, tryptophan, we have completely different protein. Yes?
If we have a deletion of 3 nucleotides, the protein will be functional or not?
If you have what?
Removal, deletion of 3 nucleotides.
Well, if it's missing amino acid, it depends where the mutation is. And it could end up changing, depends whether it's acidic protein, amino acid, if it's basic amino acid, it all depends on what changes. Okay? Here's some examples for you. And this is actually what we're going to be doing, for instance. We'll just, for instance, for TBL, I'm trying to give you examples of what kind of questions to expect. If I give you a case, in this case, for instance, sickle cell anemia, we'll discuss it in a minute. I'll say this patient had 1, 2, 3, 4, and then I'm going to ask you about biochemistry, about, okay, the mutation, how do you think this happened, why, and so on, just to see if you understand, for instance, the effect on secondary structure, tertiary structure, when you study proteins and so on. These are the kind of questions. We'll give you a scenario in TBL, and then the question is going to be about biochemistry-related questions, not about the diagnosis and so on. These are the kind of questions to expect in your TBLs and so on. Case scenario for as groups to discuss. So here's an example, sickle cell anemia. At this stage, I keep telling you guys, I'm not going to ask you about diagnosis. I don't expect you to diagnose. That's why we give you a scenario, but we don't expect you to diagnose it. We expect you to understand the basics. This is year 1. You're still not familiar with diseases and so on. When you go to 3rd year, 4th year, 5th year, then we give you the symptoms. You're supposed to diagnose and tell what's happening from our side point of view, biochemistry point of view, for instance. But don't expect that you will be asked to diagnose certain condition by giving you symptoms and so on for year 1. We'll be meeting in year 2, year 3, and so on. So we'll take it notch where we're going to be asking you about diagnosis and so on, okay? If you see that, oh, you've been asked to diagnose, challenge us. The way it works, after the final of your block or course, the batches will collect all the questions, challenges. They filter it and send it to the assessment unit, which will send it to us as well. Okay? So this is how things work. What is sickle cell anemia? I'm sure that many of us have a relative or a friend who has sickle cell anemia. It's common, especially in the Middle East. Sickle cell anemia, what happens if you look into the normal red blood cells? They're nice desks, but because of the sickle cell anemia, they start forming these sickle cells, and they aggregate, and they basically cause blockage, clotting, and this would result in ulcers, as you will see, and so on. So because of the fact that there's abnormal hemoglobin inside these RBCs, they change their shape into sickle cells. They could form some ulcers, as you see, or these are the symptoms. I'm not going to ask you about the symptoms, but look at it, and if I were you, I would just read it, because you're going to hear a lot about sickle cell anemia in the next few years. So you might know it from now. These are some symptoms associated with sickle cell anemia. Rapid heart rate. Why? I mean, if you understand the mechanism, we have clogging, we have poor oxygen delivery to the tissue. So all this would result into all these symptoms. Now, if you look into even the percentage, if I tell you 30% of Jamaican patients with sickle cell develop ulcers, 30% of the Jamaican patients will develop ulcers compared to 1% to other patients. The whole idea here is that there's genetic, mostly. You can see that because we have a mutation in one of the amino acid, as you'll see. So there's a point mutation, but it gets inherited and passed. No, there's more susceptibility, I would say, to get the ulcers. Okay? So if you look at the sickle cell anemia, and it's caused by 1 point mutation of hemoglobin B. Beta chain, where the alpha chain is unaffected. You already know the structure of hemoglobin. All what it is, we converted glutamic acid, which is acidic, which has acidic side chain, basically, to valine, which is non-polar amino acid. From glutamic to valine, all this leads to this aggregation of this hemoglobin and sickle cell anemia. 1 point mutation. Okay? So, again, I could give you a case of sickle cell and then start asking you what happens in the point mutation, how do you think this affected the secondary structures, blah, blah, blah. This is the kind of questions to expect. Okay? Another example, Huntington's disease. We'll discuss it in more detail, basically, where you can see there's a repeated trinucleotide, meaning the sequence of CAG or CAG, basically, is repeated multiple times. The CAG codes for glutamine. So you have multiple repetition of glutamine because of the fact that this sequence, the codon, the CAG, is repeated several times. And Huntington's disease is basically a neurodegenerative disease that has some problems with muscle coordination and leads to cognitive decline and dementia. This is how normal brain slice looks like, and this is how it looks like with a patient with Huntington disease. All what it is, this is normal protein, Huntington protein. This is how it looks like. This is 3D structure of it. When you have repetition of glutamine, several repetition, this allows aggregation, basically, of Huntington protein, and this would kill the neuronal cells, as simple as this. You end up from this section how it looks like normal brain, to this brain slice with Huntington disease. Simple. Repetition of few amino acids. Let's talk about cystic fibrosis. And you'll hear a lot about cystic fibrosis. You know what cystic fibrosis is by now, right? You always see it in the movies. There are so many movies where you see the mother massaging the back so that the mucus could basically allow air passage. But it's not only affecting the lungs, it affects everything. All organs are affected in cystic fibrosis. But you see the impact on the lungs, but all organs basically are affected with cystic fibrosis. What happens? We have a protein called CFTR, cystic fibrosis transmembrane conductance regulator. And there's a point mutation in it, actually deletion, not point mutation, there's a deletion of an amino acid, as you will see. And all what it is, this is an X-ray of a patient with cystic fibrosis filled with mucus. This is a receptor, actually transmembrane, basically regulator. It regulates, it's in the plasma membrane, you can see, the chloride returning back to the inside of the cell. Now, if there's a point mutation, you can see that this gate is blocked, changing the 3D structure of this gate, basically, the CFTR, and you can see that there's no more chloride coming inside the cell. And as a result, sodium stays to neutralize the charge of chloride. That's why patients with cystic fibrosis, the best diagnostic test, just the sodium chloride. They're going to have salty skins and so on, because it's excreted by the sweat and it's not returning back. Sweat gland secretes the sodium chloride and doesn't get back. That's why they have sweaty skin. And that's one of the diagnostic tests for it. But at the same time, we end up with mucus buildup outside the cell. That's why the lung is filled with mucus. Okay, what happens here with the cystic fibrosis? All what it is, the most common mutation is basically deletion of phenylalanine. So the delta F508 is the most common mutation causing cystic fibrosis. And this mutation results in the deletion of single amino acid, phenylalanine, at position 508. Now, let's move on, try to finish on time. What is needed for translation to occur? I put for you a link to a nice video. You can watch it, and it will really simplify the whole process for you of what is needed, how translation occurs. Okay? So I'll leave it up to you to look into it. One thing I want you to remember, if you're missing one amino acid, especially essential amino acid, there won't be synthesis of that protein. As simple as this. So, one thing we need to be aware of as doctors is that we have to make sure that people, whoever, if you're advising for nutrition, that you have the essential amino acid available, because if they're missing, of course, they're coming from the diet. If they're missing, basically, you're not going to have protein synthesis. It's as simple as this. If you notice that we do find supplements, essential amino acids, now we understand why, because if you're, for instance, bodybuilder, you need all the amino acid to be present for that protein to be synthesized. Otherwise, there's no synthesis. As simple as this. Now, let's talk about the translation. What do we need? The first thing, transfer RNA. Which RNA polymerase synthesizes transfer RNA? We have 1, 2, 3. We know 2 is for mRNA. 1 for ribosomes, ribosome RNA. 3 for transfer RNA. Now you know why we have 3 RNA polymerases, right? So there are around at least 50 species of transfer RNA, and of course we have 20 amino acids. So you can see that more than one transfer RNA could dock and load that amino acid. This is how transfer RNA looks like. So it's an RNA, but it has loops and has secondary structures. You can see it right there. But I want you to know this because we always ask tricky question about this. How? Why? Look into the orientation here. We have 5' end of the transfer RNA. And then we have 3' end. And that's where the amino acid is linked. Right there. Okay? So we have 5. It's an RNA after all. 5', 3'. And it is synthesized by RNA polymerase 3. And what happens? Look into this sequence. And this is the anti-codon. Anti-codon looks like what? What do you think? Template DNA, right? And it's complementary to the codon, the mRNA, right? It's complementary. This is how we look at it. Always look into orientation, 5' to 3'. It's by default 5' to 3' when we give you the sequence. Unless I give you opposite, I say this is 3', this is 5', then by default you always take it from 5' to 3'. So it's complementary. And do expect questions where I say which transfer RNA would dock with this mRNA, with this sequence. You should know. And always follow the orientation. Clear? And just to let you know, if it's something I don't say, I'm not going to ask you about it. As simple as this. And if it's something I did not explain or not talk about, you can challenge me for that. I have no problem with that. But in order for you to understand, I really strongly suggest that you read your textbook as I told you. It just explains more and better in a better way as well. I have only 40, 50 minutes. I cannot cover everything. Okay. So the binding of the transfer RNA anti-codon to the mRNA, codon follows the rules of complementary and anti-parallel binding, as we mentioned. Read 5' to 3', and so on. We have Wobble hypothesis. You don't need to know anything about it. So why am I telling you about it? Because you'll see it in your textbook, and I got so many questions about the Wobble hypothesis and so on. I don't care about it. It's a hypothesis. Okay, but what is it? In case you're curious, I'll just give it to you. Basically, you can see that this is the transfer RNA. And what happens? We have pairing, normal pairing. You will find, for instance, anti-codon, GAC, CUC. There's complementary, anti-parallel. But sometimes the last nucleotide of the mRNA can take different. As a result, we can see that the same amino acid being loaded into different codon, the wrong codon. Why? Because they say this is what they call the Wobble, allows a single transfer RNA to recognize more than one codon. I don't care about it. You're interested? In case you see it in your book or books, it's up to you. I don't care about it. So what is needed for translation? Well, we have messenger RNA. We discussed the messenger RNA. And we talked about the 5' capping, right? And it's very crucial for translation because this is how the ribosome recognizes the mRNA, and then it moves. You can see that there's a small ribosomal subunit. The movie link will give it to you really in a simple way, where you can see that the small ribosomal subunit will recognize the cap, then starts moving, and then it finds the start codon, the AUG. That's why I asked you to look into the AUG. The start codon Once it recognizes it, now the large ribosome subunit will assemble. Now we have a fully functional ribosome or polysome, and now starts moving, and you can see that we have the N end, and we already know from the proteins that we have the N and the C terminal, right? So they have N end right there coming out, and the C terminal being synthesized. So synthesis of protein goes from N to the C terminal. Now you can see the nascent polypeptide being growing, and eventually this ribosome reaches a stop codon, UGA, in this case. And what happens? We have factor, release factor, elongation release factor, ERF, comes in, and then boom, the two basically dissociate, and it stops right there. This is how translation occurs. Now, components required for translation? Well, we have the ribosomes. And we talked about the RNA polymerase that synthesizes the ribosomes, ribosomal RNA. And that is 1, right? So, what are you telling us that the ribosomes are only composed of RNA? No. It's RNA and proteins. In prokaryotes, we have, if you add these up, 55 proteins in this ribosome, the complete polysome. So what you find here, what you have here, we have two subunits, the large in the prokaryotes, 50S and the 30S. In eukaryotes, we have the 60S and 40S, the large and small. Do I need to know this? The answer is yes, because this is how most antibiotics could recognize or differentiate between humans and bacteria, and thus we can target this bacteria. So what happens? We have the 50S, 30S, and both form the full ribosome. This is subunit, first subunit, second. Both form the full ribosome. But why? They're not additives, because here we're talking about 30S sedimentation. S is a sedimentation factor, and that's affected by two things, the mass and the shape. So when we spin it, it separates accordingly. So, but it's not additive. You cannot just add 50 plus 30, 80. No, it's 70. Because S is, you don't need to know about this. But in case you wonder why it's not additive. Now, in this large subunit, the 50S, it's composed of RNA, ribosomal RNA, which we said, synthesized by RNA polymerase 1, and 34 proteins. You don't need to remember the number of proteins, but just know that each subunit is composed of RNA and proteins, and then both subunits form the complete ribosome. For eukaryotes, we have 60S, 40S. Again, we have 50 proteins, 35 proteins, and so on. You don't need to remember the number of proteins. But also, look into the RNA. For the large subunits, we have 23S, and for the small, we have the 16S. Do I need to know this? Yes. Why? Because you're going to see that which RNA would act as an enzyme, as you will see, that will form the peptide linkage. We'll discuss it. Look into here. We have 60S, 40S. We have 28S ribosomal RNA along with 50 proteins. Don't worry about the small ribosomal RNA. And then we have the 18S. So prokaryotes have different ribosomal RNA. Now, this is the large subunit. This is the small subunits. And they form the ribosome. We have 3 sites on the large subunit. These are the EPA. This is how I remember it. EPA. My wife is Heba. That's how I always remember her. Heba, EPA. EPA. E, exit. P, peptide. A, axis. meaning that you'll see that the transfer RNA will exit from the exit, E, EPA. P is the peptide formation, basically site, peptide linkage. A, site, axis, where this is where the transfer RNA gets in, okay? So let's go back to the mRNA. We talked about the Shine-Dalgarno sequence. You remember when we said the Shine-Dalgarno sequence is what? This is the site for ribosomes to bind and translate. You remember when we talked about polycistronic and monocistronic, and we said that there are multiple Shine-Dalgarno sequences, right? So the Shine-Dalgarno sequence is located between 6 to 10 bases upstream, and purine, and this is basically in this case, okay, although I'm talking about prokaryotes, you should be able to link it to the eukaryotes, as simple as this. Okay, and I'll give you some examples. So the prokaryotes, we have the Shine-Dalgarno sequence, and in the transfer RNA, it has a sequence complementary to this Shine-Dalgarno sequence. As simple as this. Purine, purine, purine. This is a repetition of purines, right? So the 16S ribosomal RNA, as I indicated for you, the small subunit, will bind to this Shine-Dalgarno sequence, and it will facilitate the initiation of protein synthesis at the 5' end of the mRNA and 3' end of the 16S. They're complementary. So the 5' here, complementary to the 3' end of the 16S ribosomal RNA, which can form complementary base pairing, facilitating the positioning of the small subunit, ribosomal subunit, and then the large subunits. We have the start codon, and we have the termination codon and poly A. So the initiating codon is AUG, right? Codes for methionine. So methionine is the first amino acid to start the protein synthesis, as simple as this. Now, the charge initiator transfer RNA enters the P site only for the first amino acid. Usually it enters from the A, EPA, EPA, A axis. But for the methionine, the first one, it is methionine, if it's part of the initiation codon, methionine will get directly into the P site. And you'll see why. Now let's say this is the translation of an mRNA. You have the mRNA 5' to 3' right there. The ribosomes assemble and starts moving along the mRNA. If you notice here, we have the polypeptide being synthesized, right? It's basically attached to the transfer RNA. Now, the new transfer RNA with the amino acid that corresponds to the codon here will enter through the A site axis. Okay? Now we do have utilized energy, GTP, and what happens? Notice that the nascent polypeptide being synthesized now is forming a peptide linkage to the one in the A. And this is why we call how could transfer RNA act as an enzyme? Enzymes are proteins. In this case, the ribozyme is basically a transfer RNA acting as an enzyme, as a protein, functioning as a protein. And the function of it, it's called peptidyl transferase. And this belongs to the 23S ribosomal RNA. Is it eukaryotes or prokaryotes? Prokaryotes. What about the eukaryotes? 28S has that function. Everything will be comparable. So the peptidyl transferase basically is the 23S ribosomal RNA. It's a ribozyme. It will form a peptide linkage. And notice here now, the polypeptide being synthesized, it's in the A site. The ribosome moves after that. And you can see now, the ribosome moves to the right, to synthesize from 5' to 3'. And what happens? Now the empty transfer RNA exit through the E. And everything now is in the P site. And this continues and so on. The movie will explain it to you in more details. Elongation. Elongation, now we continue with the synthesis, the ribosomes, they continue with the synthesis. And finally, when we reach to the termination codon, everything I just skipped, I already talked to you about it. In case you missed it, it's already written for you. We reach the termination codon. What happens with the termination codon? I told you, there's a factor called ERF, as an example. Elongation release factor binds, and this will cause the whole thing to basically disassemble. Now, do you think the termination codon, there's an amino acid coding for the termination codon? No, it does not get read. So if I give you 9 nucleotides as a question, and I say, how many amino acids are going to be formed? 9 nucleotides. 9. And then you see that the last one is termination codon. 2, 3, and the last one does not code for an amino acid. This is how simple it is. I can't believe me, I have a million ways of just to trick you unless you understand the concept. I don't ask you what is the termination codon, but if I give you such question, you should understand what's the termination codon in order for you to be able to answer this. Polyribosomes, meaning that we could have multiple methionine and more than ribosomes synthesizing that, basically protein, because it's a long mRNA to speed up things. Protein targeting, I'm not sure if I can finish because I really want to I need to ask for another 15 minutes from another session, not today. I'll try and get 10-15 minutes afterward because I really want to spend some time with this co-post, translation and so on. So we'll stop here. I'll take that attendance because I know you're tired. We'll give you a break.